We shall start this class with a question. Can humans produce vitamin C? We shall discuss the answer for this question with reason by the end of this class. Hello students. In our previous classes on carbohydrate metabolism, we have already discussed about the pathways involving oxidation of glucose, like glycolysis, aerobic and anaerobic, glycolysis in cancer cells, glycolysis in RBCs, that is Rappaport-Lubering shunt, HMP shunt. The topic of today's class is uronic acid pathway. It is also an alternate pathway for oxidation of glucose. The pathway is named as uronic acid pathway because the product of this pathway is uronic acid. How is uronic acid produced? By oxidation of carbohydrates. Thus, oxidation of carbohydrates forms the corresponding uronic acid. Examples: glucose on oxidation forms glucuronic acid. Galactose on oxidation forms galacturonic acid. Mannose on oxidation forms mannuronic acid. Thus, uronic acid pathway is for oxidation of glucose. What is the other pathway for oxidation of glucose? Yes, it's glycolysis. Thus, uronic acid pathway is considered as an alternative pathway for oxidation of glucose. What are the end products of glycolysis? It's ATPs and pyruvate or lactate. But here in uronic acid pathway, there is no ATP generation and there is no pyruvate or lactate production. So what are the products of uronic acid pathway? UDP glucose, glucuronic acid, pentose sugars, ascorbic acid or vitamin C. But this ascorbic acid cannot be produced in humans. What is the site of uronic acid pathway? It is liver cytosol. The major product of uronic acid pathway is glucuronic acid. Now we shall discuss about the applications of glucuronic acid. There are two major applications of glucuronic acid. The first one is detoxification. This glucuronic acid is a strongly polar substance. Thus, it helps in conjugation reactions. Glucuronic acid, when it gets conjugated with a water insoluble substance, it will make that water insoluble substance to water soluble substance. Thus, it can help in renal excretion. So, the water insoluble substance can also get excreted in urine when it gets conjugated with glucuronic acid. Examples: conjugation of bilirubin, drugs, and steroids. The next important application is synthesis of glycosaminoglycans, that is GAGs. This glucuronic acid is a component of chondroitin sulfate and hyaluronic acid. And this glucuronic acid also helps in synthesis of L-iduronic acid. L-iduronic acid is an epimer of glucuronic acid. This L-iduronic acid is a component of heparan sulfate and dermatan sulfate. Now we shall see the steps of glucuronic acid pathway. The substrate of glucuronic acid pathway is glucose 6-phosphate. How is glucose 6-phosphate produced? Glucose 6-phosphate is produced from glucose by the enzyme hexokinase. This glucose 6 phosphate in the presence of phosphoglucomutase gets converted to glucose 1 phosphate. Then this glucose 1 phosphate reacts with UTP, that is uridine triphosphate, to produce UDP glucose, that is uridine diphosphate glucose, in the presence of enzyme UDP glucose pyrophosphorylase. Now the first product of glucuronic acid pathway is produced. This UDP glucose in the presence of NAD dependent UDP glucose dehydrogenase gets oxidized into UDP glucuronic acid. UDP glucuronic acid in the presence of UDP glucuronidase, UDP is removed to form D-glucuronic acid. This is the next product of uronic acid pathway. This D-glucuronic acid in the presence of NADPH dependent glucuronic acid reductase produces L-gulonate. This L-gulonate is the precursor of vitamin C or ascorbic acid in the presence of enzyme L-gulonolactone oxidase. In humans and other primates, this enzyme is absent. Hence, vitamin C or ascorbic acid cannot be synthesized in humans and other primates and also guinea pigs. This L-gulonate undergoes oxidative decarboxylation in the presence of L-gulonolactone dehydrogenase to produce L-xylulose. This xylulose is a pentose sugar which is another product of glucuronic acid pathway. This L-xylulose gets reduced to xylitol in the presence of NADPH dependent xylulose reductase. This xylitol gets oxidized to D-xylulose in the presence of NAD plus dependent xylitol dehydrogenase. Now D-xylulose gets phosphorylated to xylulose 5-phosphate in the presence of xylulose kinase. This xylulose 5-phosphate gets further metabolized in HMP shunt and leads to formation of glucose. Now we shall see about a disorder associated with uronic acid pathway defect, that is essential pentosuria. Essential pentosuria is a component of Garrott's tetrad. Tetra means 4. So Garrott's tetrad includes 4 inborn errors of metabolism. We can remember these inborn errors with a mnemonic PACA, PACA. P for pentosuria, that is essential pentosuria, A for albinism, C for cystinuria, and another A for alkaptonuria. Essential pentosuria is an autosomal recessive disorder. Incidence is 1 in 2500 births. Enzyme deficiency in this disorder is xylitol dehydrogenase or xylulose reductase. What are the functions of these enzymes? These enzymes help in metabolism of xylulose. If these enzymes are deficient, then xylulose gets accumulated inside our body. If xylulose gets accumulated inside our body, it can get excreted in urine. But these patients are usually asymptomatic. Only during consumption of large amount of pear fruits. Since pear fruits are rich in pentoses, there will be alimentary pentosuria. If these patients consume drugs like barbiturates, aminopyrin or antipyrin, these drugs will induce uronic acid pathway and it will induce xylulose in these patients. So when these patients consume these drugs, there will be increased excretion of xylulose in pentosuric patients. Investigations include, since xylulose is getting excreted in urine, we can check for increased urine xylulose levels. As xylulose is a reducing sugar, Benedict's test will show positive in these patients. And Bial's test, which is for pentosis, also shows positive in these patients. We can also do paper chromatography, a technique used for separation of carbohydrates for diagnosing this condition. Treatment: this condition does not produce any harm usually, but it should be differentiated from diabetes mellitus, because Benedict's test, it will show positive both for this condition, essential pentosuria, and also for diabetes mellitus. So this condition should be differentiated from diabetes mellitus. Thank you.